So today we're going to talk about feedback mechanisms and how they're used within an organism to maintain dynamic homeostasis. Now, homeostasis refers to maintaining aspects of the organism's internal environment within parameters that allow the organism to maintain normal functioning. So those parameters can be things like body temperature, blood sugar, and so on. So in order to really understand this, we're going to need to look at an example. The example we're going to look at today is regulation of energy metabolism. The primary source of energy in our bodies is glucose. Now, glucose is particularly important because there are some tissues in our body that can only use glucose for energy production, and there are others that are very picky and would really prefer to use glucose whenever possible, though they have a few other options. So you should think of glucose as our default energy source. Our other major energy source is fat. We use fat because it's very good for energy storage. It's more energy dense than glucose, which basically means the same amount of energy takes less space to store. Now in humans, we can easily take the energy in glucose and turn it into fat. However, we can't do much in terms of going the other way. We can't get most of the energy out of fat and turn it into energy in the form of glucose. However, we said that there are some tissues that really want to use glucose if at all possible, and there are some tissues that can only use glucose. So our body has two solutions for this. First of all, we can actually generate some glucose from protein by breaking it into its constituent amino acids, some of which can be used to produce glucose. Now that said, fat isn't going to be a very good storage source if we can't use it to supply energy to some of our most energy-hungry tissues, such as the brain, which usually uses glucose. So we actually have another solution, which is to turn fat into something that the brain can use, called ketone bodies. Given this information, we can now look at the two major goals of energy metabolism regulation. So the first goal pertains to when we're using primarily glucose as our energy source. And in this case, we need to manage glucose levels because when glucose levels get too high, they're toxic. The major hormone that is responsible for this job is insulin. The second thing we need to do pertains largely to that fat burning mode. And when we're burning mostly fat, remember that some tissues still need glucose and some tissues need the fat processed a little bit in advance for them into ketone bodies. So we have to ensure that we supply those picky tissues. The hormone responsible for managing this is glucagon. Now, biochemical pathways can get a little bit confusing, but hang in there. We're going to keep it as simple as we can by simplifying the pathways and just focusing on key steps for regulation, rather than looking at every detail in the pathways. So let's start by looking at what happens when glucose is our primary source of energy being used by most of the cells in the body. Under these circumstances, insulin is running the show. Insulin's being secreted anytime sugar is plentiful and therefore blood sugar is high. Insulin allows cell types that can use multiple types of energy to pull in glucose from the blood. Without insulin giving the signal that this is okay to do, cell types like muscle cells that can use multiple sources of energy will leave the glucose in the blood for cell types that need it more. Once glucose is inside the cell, we can start putting it through the pathways to generate energy. The first step for that is going from glucose to pyruvate. This process is called glycolysis. Glucose is a 6-carbon sugar, pyruvate is 3. So we're cutting it in half effectively, and this process generates a little bit of energy. I should note that that first step happens in the cytoplasm of the cell, whereas the next step happens in the mitochondria. So to keep clear where we are in the cell, I'm going to draw a line here. After glycolysis is completed and we have pyruvate gets moved into the mitochondria, where it is turned into acetyl-CoA. Acetyl-CoA is only 2 carbons, so if you're wondering where that carbon goes, it gets released as carbon dioxide, and that step also generates a little bit of energy. Acetyl-CoA is now ready to enter into the citric acid cycle. It does this by combining with a 4-carbon compound, oxaloacetate, to form a 6-carbon compound called citrate. Now for today's lesson, we don't really need to get into the details of the citric acid cycle. So what you really need to know is that Citrate goes through a number of steps to reform oxaloacetate, and in the process, harvesting the energy stored in those 2 carbons of acetyl-CoA as ATP and NADH, and releasing those 2 carbons as carbon dioxide. So under the conditions where we're using glucose as our primary energy source, that's usually because glucose is fairly plentiful. So we can also have 2 other processes going on to promote energy storage. First off, we have the production of glycogen. This can happen in lots of tissue types, but the liver in particular stores a lot of glycogen. Most of the glycogen stores in your body are in the liver. In the types of cells that store fat, so primarily fat cells, we can take citrate out of the citric acid cycle and out of the mitochondria, and we can use citrate to produce fatty acids, which of course can be used to make fat. So these are the pathways that are running when glucose is plentiful. And remember, under these conditions, we have insulin being secreted. So we want to remember that insulin is responsible for promoting the uptake of glucose from the blood, promoting the production of glycogen in the liver, promoting glycolysis and the feeding of the products of glycolysis into the citric acid cycle in all of the tissues in the body, and producing fat in the fat cells. So now we're going to look at what happens when we're using fat as our primary energy source. Fat in fat cells is lysed to release free fatty acids, which are released into the blood. These are then taken up by cells as a source of energy. Once they're in the cell, the fatty acids can be transported into the mitochondria. So let's draw that line again, where they are converted into our old friend, acetyl-CoA. And as we know, acetyl-CoA is ready to be fed right into the citric acid cycle. So it combines with oxaloacetate, forming citrate, and the citric acid cycle can run as usual, producing energy. So this works in all of the cells that can use fatty acids, but what about all the cells that can't? The liver is in charge of making sure the cell types that can't use fatty acids have what they need. So in the liver, these pathways look a little bit different. Instead of letting the oxaloacetate get fed into the citric acid cycle to make citrate, oxaloacetate is pulled out of the citric acid cycle and out of the mitochondria, where it's used to go backwards to pyruvate. Pyruvate can then be turned into glucose. And that glucose can get released into the bloodstream to supply cells that require it. Additionally, remember that the liver stores glycogen. So as long as there are glycogen stores, glycogen can be broken down into glucose. So the liver has 2 places to get glucose from. Eventually, we're going to use up all the oxaloacetate that was in the cell, and we can't make any more from citrate because we are not running the full citric acid cycle. However, we can run part of the citric acid cycle using protein to come into the cycle midway, so that can continue to supply oxaloacetate for a while to make more glucose. So the acetyl-CoA is still here because there's plenty of fatty acids in the cell, so acetyl-CoA is still being made. And though the liver's working pretty hard to make glucose, there's a limit to how much glucose those systems can make. So to ensure that the brain, a really energy-hungry tissue, doesn't starve, we produce from acetyl-CoA ketone bodies. Just as we noted for insulin, which of these processes was driven by insulin, we're going to do the same for glucagon, which is the hormone present when blood sugar is low. Glucagon directly promotes the lysis of glycogen to produce glucose, the production of glucose from pyruvate, the transport of fatty acids into the mitochondria, and the productions of ketone bodies from acetyl-CoA.